Well, thank you for a very generous introduction. I also like to thank Dr. Legato and her team for organizing this wonderful meeting in a wonderful place. I'm really grateful to be here and talking about our work. So what I plan to do in the next 25 minutes or 30 minutes is to give you a different flavor of the epigenetic regulation by mitochondria. And particularly, what I'm going to be talking about is the mitochondria as the epigenetic regulator of reproductive aging. So we come in the picture looking at the mitochondria from the point of view where mitochondria are described as the main hallmark of aging. There are 9 different hallmarks of aging, as you can see on this slide, but mitochondria appears to be the one hallmark which connects to the many other hallmarks in the process. So here, what you're looking at is uh, the mitochondrial dysfunction leading to many of these hallmarks. In fact, our laboratory has contributed over the years where we showed that the mitochondrial dysfunction leads to cellular senescence, it leads to altered intracellular communication, genomic instability, and the telomere attrition. We have shown that the mitochondria can in fact regulate telomere as well. So what we wanted to do is to focus on the mitochondrial dysfunction for looking at the aging in the whole animal, and then later on looking at the aging of the different organs in a specific way. So when it comes to mitochondria, it is important to realize that the mitochondria are maternally inherited. When the fertilization takes place, the sperm mitochondria are degraded by the egg, and only the mother's mitochondria are transferred. Except there's one exception in Denmark. This individual has the father's mitochondria to some extent, but he's healthy, no problem. So when we think about the mitochondria, we know that mitochondria are the powerhouse of the cell, but mitochondria are more than the powerhouse of the cell. We have been focusing on describing the different roles of mitochondria in the cell. And when we talk about the mitochondrial role in powerhouse, what we do know that the mitochondrial DNA encodes the 13 proteins. So for example, here you have the mitochondrial DNA encoding 7 proteins which are part of the complex 1, and the 35 subunits are encoded by the nuclear DNA. So what happens that these 35 subunits are synthesized in the cytoplasm, transported into the mitochondria, and together with the 7 subunit encoded by the mitochondrial DNA, it becomes the complex 1. And there are many different factors which are involved in the assembly of the complex 1 or different complexes. Complex 2 is exclusively nuclear, and so you also see the complex 3, 4, and 5. You have various different subunits encoded by the mitochondrial DNA and the nuclear DNA. So what it tells us is that there's a constant crosstalk between the mitochondrial genome and the nuclear genome. And obviously this is going to have a cellular effect and the epigenetic effect which we discovered, and I'll describe to you in a minute. But beside that, what happens is that mitochondria contain close to about 2,000 different proteins whose function is somewhat unknown. And so we have to think about mitochondria beyond the ATP production. And what I've done actually in this particular slide is to give you the summary of what we actually published recently, that the mitochondria are involved, as I mentioned, in ATP production, but what appears to be that there is different steps in the information transfer by mitochondria within the cell and outside the cell. So you have intracellular transformation where nucleus signals to the mitochondria, so you have anterogate response. Then you have a retrograde response when the mitochondria don't work properly. They send the signal, and that nucleus takes care of it. Otherwise, there is self-fate decision has to be made. And there we have shown the epigenetic regulation and the genetic regulation by the transient or permanent dysfunction of mitochondria. Then you also see that there is extracellular transfer, information transfer, where ATP is leaked out when mitochondria have enough ATP and they don't need it. The ATP is leaked out in the outside the cell, and then through the purogenic receptors, so there are 7 of these purogenic receptor, ATP is taken back, and that signal is also very complicated and involved. Then you have the intercellular information transfer, where mitochondria itself can move from one cell to other. Beside the direct transfer of mitochondria, you also have the mitochondria-derived peptides that can also move, also find mitochondria in the exosomes, and the mitochondria-derived vesicles. So these mitochondria throw out small vesicles. They contain different sets of proteins, and they talk to in the cell with the endoplasmic reticulum and others. So it's like a different cargo having various different sets of protein. But what also interesting and is worth noting is that neutrophil, extracellular tract, contains the mitochondrial DNA as well as eosinophils. So why in the world for immunogenic response to kill the bacteria, the mitochondria are outside the cell, the extracellular tract contains the mitochondrial DNA and kill the bacteria. In addition to that, mitochondria are a hub for innate and adaptive immune response. So what I think time to emphasize here is that when we think about ATP production only, we have to go beyond and looking at where mitochondria are involved in all these functions of mitochondria. So, and something which has been hidden in the literature is that the mitochondria are also critical organelle for endocrine health. So the progesterone, the mineralocorticoids, glucocorticoids, androgen, and estrogen, they all go through the pathway which are involved in mitochondria. And our laboratory has shown that the androgen receptor as well as the estrogen receptor also localizes to mitochondria, and we are trying to figure out what that function means in terms of the localization. And when we look at the sex difference in the mitochondrial function, women seem to do better than the men. They respire, their respiration of the mitochondria is much better than the men. And correlation to that is the women seem to live longer in terms of the aging than the men. But unfortunately, I have a slide I'll show you, is that women's health span or their later life is not disease-free. So here is what I just talked about it, that when we look at the sex differences, the disease incidence, you find that in women, the autoimmune diseases are much more frequent than in men. And you also find cancers is more prevalent in men, but the neurodegenerative diseases are more common in women. So, and here you see more in detail in the terms of the cancer incidence. So why women have higher incidence of thyroid cancer compared to men, we don't know. There's something we are working on trying to figure out. So when we talk about the complexity of the mitochondrial function, we also have to take into account the mitochondrial number varies from tissue to tissue. So mitochondria can be anywhere between 200,000 in the grown oocyte to 1,000 in the primordial oocyte. And this is the mouse data. When we look at the human numbers, there's a 5 million mitochondria human oocyte. So obviously, and those mitochondrial function is I would say unknown, because obviously ATP production is one of them, but what else they are doing in terms of the oocyte function is still work need to be explored. So mitochondria play a critical role in ovarian function and aging, and that's I'm going to show you the data we have. But what is also important to realize that 1 in 100 women suffer from premature ovarian failure, and this appears to be very similar to what we call early menopause. So what you see is the symptoms are vaginal atrophy, menopause, infertility, the cognition issues, bone disease, metabolic and cardiovascular disease, and overall reduced life expectancy. So what we were looking for is that given that the role of mitochondria and the number of mitochondria, which are very many in oocyte, how we can look for and identify the mitochondrial role in ovarian function, aging, and menopause. What turns out to be that in the literature by this group here, they pointed out that there are close to about 8 different genes which are involved in premature ovarian failure. So here what you have is the polymerase gamma, which is the protein involved in the mitochondrial DNA replication. Twinkle is one of the helicase that's also involved in mitochondrial DNA replication. And then what you find that these proteins also, they carry on the transcription, the twinkle helicase. Then there are these proteins which are involved in the translation. And then you have these proteins in the tRNA synthesis, and that this clip is involved in the processing of the mitochondrial peptides. So obviously it tells you, and 10 to 20% of the PIU cases are due to mutation in a gene impacting mitochondrial function. And unfortunately, the only option for women is the egg transplantation. And I want to emphasize here that this, and the POI is, as I mentioned, 1 in 100. That amounts to close to about 35 million in a conservative estimate around the world. And we have nothing to help these women accept that egg transfer. In some cases, the egg transfer is prohibited. For example, in Germany, you cannot have egg donations. So there's a lot of work need to be done. So we developed an animal model, and when it came to looking for animal model, obviously we were interested in overall aging of the mice when the mitochondrial function disrupted. When we particularly looking for the mouse model or model for understanding ovarian aging, there appears to be only one, and which is very, I would say, the toxicology model, where what you have is this compound VCD. You expose the mice or rat to about a month, and then these mice show the premature ovarian failure or premature menopause. So obviously this is a very different set of model one can use. So what we decided to do is look for polymerase gamma as a target gene where we can disrupt mitochondrial function at will in the whole animal, so we can study the aging of the different organs, but also the ovarian aging in general. So, and this also fits in with what I've described, that when you look at the mitochondrial DNA content in premature ovarian aging, so here you have the normal, this is a study done by this group here, that where you see in the CPM, which is the old and the physiological menopause beyond 48 years of age, you see the mitochondrial DNA is down. Then you look at the poor responders, and then you look at the premature ovarian failure, the mitochondrial DNA content is low. So it gave us the idea that perhaps that the genes which are involved in the mitochondrial DNA replication are the key player, or at least one of the player in premature ovarian failure. So what we decided to do is look at the polymerase gamma, identify a region, and in fact you see this region is conserved. So the polymerase gamma is made up of three domains, is the exonuclease domain, the linker domain, and the polymerase domain. So the mutation in the exonuclease domain would lead to mutation in the mitochondrial DNA. Mutation in the polymerase domain will lead to the depletion of the mitochondrial DNA, as I described in the previous slide. So what we decided to do is develop a mouse model where we can take advantage of 1135 mutation, which we had in the laboratory and in cell culture model, and identified that this is a dominant negative mutation. We hook that to the GFP on the bicyst running promoter, so the wherever 1135 polymerase gamma will be expressed, what we will see is the GFP is also expressed. So in doing so, what we can do is go after identify any of the cell type we want and study those cell type at the single cell level or organ level. Then we can take this mouse 1, we cross it with the CMV RTTA, which is a constitutive promoter. So what happens that when we make mice 3, that will express the polymerase gamma dominant negative in the whole animal. And now we can take this mice here and cross it with any of the RTTA. There are close to about 150 different RTTA mice, tissue specific, and we can knock down the mitochondrial function at will. This is basically the confirmation that we in fact created that mice. This is the characterization of the mice, where we looked at by RT-PCR. So we're calling it the mitochondrial DNA depleter. So we looked at by RT-PCR the genes encoded by the mitochondrial DNA. So you have COX-2, ND1, you see they were downregulated. We looked for the OXPHOS subunit expression, which are encoded by the nuclear DNA. Here you see those complexes were down. And we also look at the super complexes. The thing I didn't describe to you is that when we see the complex 1, 2, 3, and 4, I showed you in the linear fashion. In fact, that is not always the case. What can happen, the complex 1 can get together with complex 3 and 4, and that will form a super complex. So that complex was also destabilized, and so was the other complexes as well. But at the end of the day, we look for the effect of these, looking at the enzymatic activity, and what we find that the complex 1, 2, 3, 4, and 5, they were all affected, and they had a reduced activity. So what turns out to be, we thought, well, when we deplete the mitochondrial DNA, these mice will die of cardiac problem or brain problem. But turns out that first phenotype we saw in 100% of the mice, that they show wrinkles and loss of hair very, very quickly. Within a couple of days. And that got us excited, so we had a bit of a sidetrack to figure out what this means. And I'm going to show you a couple slides. So this also got the attention of the world because although it's done in mice, I told the reporters and people who interviewed us that this is mice, not humans, but they ran with the story. And as you can see, this all over and actually quite interesting in many of the women's magazine, it was like, okay, we found the fountain of youth, and we can do anything we want. But that's not the case. We are working through UA Biosciences, the startup company we have formed. Be looking at some of these aspects of it, and obviously the loss of hair and wrinkle formation is also associated with chemotherapy-induced cancers, or sorry, chemotherapy-induced problems. So what we do know, the skin mirrors the aging of the other organs. So when we see that these mice show wrinkles and they show hair loss, and that phenotypically we can target and look for other organs. The one we looked at, obviously for presentation here, is the reproductive aging. And what I'm showing you here is the summary of what we have found. So what we found was the uterine and vaginal atrophy in these mice. And there was a premature onset of abnormal estrous cycle. There were fewer developing follicles. There was increased lipid-filled stromal cells, which led to the inflammation. The anti-mullerian hormone was prematurely diminished, and these mice eventually showed infertility at a young age. So I'm going to show you some of the slides, the data slides here. So here you're looking at the uterus of the wild-type mice. And here you're looking at the depleter mice, and that's showing the H&E staining. And turns out that at time point 4 to 12 months, we see that the weight of the uterus was low in the depleter mice. We looked at the vaginal atrophy. Looking at the epithelium here, the epithelium was reduced in the mitochondrial DNA depleter mice compared to the wild type. Then we looked at the estrous cycle. So the estrous cycle in the depleter mice was either prolonged or absent altogether, clearly telling us that there's an issue with the ovarian function. And then this is the depleter mice. These mice do not reproduce. They have fertility issues. So what we thought, well, we can do is, and this is a cell culture experiment we had done in the context where we can, because it's a conditional expression of the POLG, polymerase gamma mutant, we can restore mitochondrial function at will. So we can take away doxycycline from the chow, and what will happen that the mitochondrial DNA content will come back to it. So the approaches we have taken is that one, using the polymerase gamma 1135, what we can also do in cells, and in fact we can do in animals too, but that is going to be lethal, that we can completely deplete mitochondrial DNA altogether. So these cells are called row 0, devoid of the mitochondrial DNA, and now we can take anybody's mitochondria. So it gives us the advantage of studying different mitochondria from different diversity, different groups, ethnic groups, to identify their role and their effect on genetics and epigenetics in the nuclear genome. So what I'm showing you here is some cell line studies where we depleted it and then we put mitochondria back into it. These cells require uridine and pyruvate for growth. Without that, they die. So we employed a method where one of my colleagues at Roswell Park, he invented the restriction landmark genomic scanning, where we can actually scan the whole genome for epigenetic changes, for methylation. And what he found, what he utilized was the NOT1 as a landmark enzyme. So you see there are a lot of C's and G's here. And if the NOT1 does not cut, that means the CpG is methylated, but the absence of the RLGS spot will tell it that the gene is methylated. So here, that's what it looks like in two-dimensional gel electrophoresis. He meticulously have mapped that, these positions where we know where exactly in the genome the methylation takes place. So first we look at the proof of principle, whether by depletion of the mitochondrial DNA, whether we see the epigenetic changes in the nuclear genome. So A means absent, that means is methylated. P means present, is unmethylated. So the spot you will see which is present is unmethylated. So we created different sets of cell line, 3 different cell lines, where we see you see the unmethylated. When you deplete the mitochondrial DNA, what you find that this becomes absent, meaning it's methylated. You see the similar phenomena in other cell line and the third cell line here. We can quantitatively measure the methylation pattern in these promoters. And then what we did was ask the question, what happens if we put the mitochondria back into it? And this experiment was driven by, and the motivation was that in several years ago, there was a group of fertility clinicians in New Jersey and around the world. They transfer the cytoplasm from a healthy donor into what they describe as UPAS women who do not conceive. And in that process, 22 children were born around the world. But what they forgot when they were transferring cytoplasm from the healthy donor, which is 20-year-old woman, the 2 things. One, as I mentioned, the oocyte contains 5 million mitochondria. The 20% of 5 million is a hell lot of mitochondria they were transferring. We had a call with them. We wrote an article in Nature trying to figure out what it means. But what was complicated was that it in fact was a 3-parent baby. So when they transfer the cytoplasm, which contains the mitochondria, from a healthy donor, so that the children who were born contain the mitochondrial DNA from a healthy donor, the mother, and the father. Father obviously did not contain, but there were 3 different genetically distinct children who were born. So the question was, by the transfer of mitochondria, what does it do? There was no follow-up on these studies on that. It went to FDA. They are not allowed in United States, but in other countries, there are some mix and match with the technology you can use. So this experiment was driven by, with us, that, well, what happens if we transfer just in cells? Because there was no mice experiment, no cell experiment at all, but this human experiment was done. So what we thought what we can do is obviously where we can transfer the mitochondria, but what we can also do is by, as I mentioned, deplete and replete mitochondrial DNA content by the method I just described, where we can take away doxycycline from the cells or the chow, and it will deplete mitochondrial DNA. And what we find was that a large number of microRNA were associated with mitochondrial dysfunction or restoration of mitochondrial function. So here you see in day 5 where we show that there's mitochondrial DNA depletion does occur. There's a large number of these methylation of the microRNA. And then what we find that the day 12, they seem to get to close together, and it seems like the methylation pattern is gone. So what I want to show you is that how do we have identified, and restoration means that is mostly driven by the epigenetic changes. So I'm going to show you this slide here where we looked at the cells which were devoid of mitochondrial DNA, where we put mitochondria back into it, and you see that these 2 of these microRNA were upregulated and 4 of these were downregulated. We focused on 663, which because not much was known, and what we found was that whether we use the genetic approach, so for example, here we depleted the mitochondrial DNA, we put mitochondria back, this was epigenetic regulated by methylation, and whether we take the primary or the mature microRNA, both had the same pattern. In addition, what we find that this, beside the genetic approach, if we use the inhibitors of the mitochondrial respiration, whether it's rotenone or antimycin A or potassium cyanide, we see the same pattern. So 663 seems to be an important player in this phenomena. We looked at the quantitative way, what it means in terms of the epigenetic changes and particularly methylation. What I want to point out here, when we use the rotenone here, the microRNA expression goes down. When we add the azacitidine, it goes back again. And this was driven by increase in the DNA methyltransferase activity. So directly, this particular pathway is driven by the methyltransferase. We looked at, so you look at the epigenetic changes driven by the microRNA 663, we wanted to know whether this 663 or any other microRNA function as a mitochondrial checkpoint. So in other words, the job of this particular microRNA is to restore mitochondrial function and what it means when it goes down. So we looked at different gene expressions of different complexes. So here we're showing you complex 1, 2, 3, and 4. And you see that in both of these cases where we use anti-microRNA 63, this goes down. We looked at the assembly factors, which I mentioned early on, that it involved in the assembly of those complexes. What we find that the assembly factors also goes down, clearly saying the 663 regulating not only subunit gene expression, but the assembly as well. And here we're showing you the data that when you don't assemble, you're not going to make the complex form, and those are going to leading into the mitochondrial inactivity. So here is what you see, that the complex 1, 3, and 4 are gone. This is reduced, and this is completely gone. But what turned out to be that the 663 appears to directly target one of the, at least that's what we have identified, one of them, but there could be many. The assembly factor, which is UQCC2. I know that the name gets complicated, but this is a direct target of the 663. And this particular 663 is involved in the complex 3 assembly. So what it's telling us is that, so I want to show you that the effect of that is obviously at the end is the reduction in the enzymatic activity. So what I'm showing you here is that the 663, the normal function is to maintain the OXPHOS subunit expression, assembly factors, supercomplexes, enzymatic activity, but the reduction in that will lead to the reduction in those expression, and the 63 promoter methylation and demethylation drives that phenomena. So, and something so this work we started many years ago, as I mentioned, because of the UPA's story. In 2008, our group was the first one to show that the mitochondria function as a regulator of epigenetic changes in the nuclear genome. Now, since then, I am trying to do catch up because the field has exploded. The names are changed. It's called the metabolic regulation of epigenetic changes and so forth. But we are catching on in terms of looking at in whole animal and the mice to figure out. I think I've already described that. So going forward, what we want to know and what we want to do is, can we prevent, delay, or undo ovarian aging in humans? So we're using our platform and AI. We're looking at different FDA-approved drugs which we can repurpose in terms of restoring mitochondrial function in the mice we're working with, but we're also looking at natural products and many other ways to figure out whether this can be done. Lastly, I want to acknowledge the people in the lab. Jasmine Chiang is an OBGYN fellow. She's moved on and has her own practice. Noha Ahmed is a MD PhD dermatologist, and Bhupender Singh was the one who helped us develop the mice model. And I'll stop here.